In this video, we'll be looking at the concept of protein synthesis. In class, we covered this in 3 separate lectures, and this will be just one video over the whole topic in order to provide some amount of continuity. I will show you where the breaks are for the classroom lectures. A couple of things to start off the conversation about protein synthesis, one of those being this idea of an amino acid. This is what proteins are made out of. And an important part of protein synthesis is making sure that the amino acids are brought to the site of protein synthesis, which is the ribosome, and those amino acids being joined together through peptide bonds. Remember how that is made. A couple of more names to throw in there. George Beadle and Edward Tatum were popular in the 30s for developing this idea of one gene equals one enzyme. Basically the idea that a gene has the genetic information for making an enzyme. This is later changed to one gene equals one polypeptide, meaning that basically your DNA codes for proteins, which is what we have been talking about for a long time. How is that done? Well, again, here's your DNA, but also one of the things that we're going to be talking about here is RNA as well. DNA and RNA are different from one another in just a couple of things. Actually, 3 factors that we're going to look at that make them different. One of them is the sugar. For DNA, deoxyribose is the sugar. For RNA, ribose is the sugar. For RNA, instead of thymine, RNA contains uracil, like you see here in this picture. And RNA is also single-stranded, whereas DNA is double-stranded. As we are talking about protein synthesis is made up of 2 main stages: transcription and translation. That transcription and translation is going to be very similar between prokaryotes and eukaryotes. We will talk more about those differences later, but for now, just understand that transcription, translation make up this process called protein synthesis. And what is transcription and translation? Well, transcription is when you start with a DNA strand. Notice here you have your DNA, A, C, C, so forth. The act of transcription takes that DNA strand, reads it, and from it produces the messenger RNA. It doesn't convert the DNA into messenger RNA. The DNA stays there, but it creates a copy of the DNA, and the copy of the DNA is that messenger RNA. So transcription, you go from DNA to RNA, and then translation, you'll see it takes that messenger RNA, and from it reads the messenger RNA and produces amino acids. And those amino acids, in turn, are what make up your protein. Well, how does it do that? What does it get? How do you go from messenger RNA to protein? Well, this guy discovered what is called the codon chart in the when did he do this? It's been a while. 1960s. He discovered the codon chart. And what is a codon? Well, you can see it here on this picture. Codons are just 3 base units of RNA. UGG is a codon, and it codes for something. And here's what it codes for. UGG codes for tryptophan, as you can see here. And you don't have to know the names of the amino acids. But each 3 base unit codes for a particular amino acid. And so as you go through this process, the messenger RNA is made. That messenger RNA contains 3 base units. Each one of those codes for a particular amino acid. There are some specific ones that I'll point out. AUG is the start codon. It also codes for an amino acid. It codes for methionine, but it is the start codon. It is always the first 3 letters in the code. And then there are 3 codons UAA, UAG, and UGA that code for stop. Stop is not an amino acid, but stop does what you think it does. It tells the process to stop the protein synthesis. Now, I will often refer to those codons as reading frames. It's just a kind of like we read in sentences or words. This protein synthesis is read in these codons. This is a reading frame that it's read in. Now, we're going to look at the process of transcription first. Transcription is again, don't forget what is transcription. Taking a DNA and from it making a messenger RNA. And that transcription takes place in 3 stages: initiation, elongation, and termination. Well, the initiation part of transcription is what I refer to as building the factory. On this particular strand of DNA, there's a gene. You can see it calls it here the template DNA strand, whatever. Every gene has a promoter section on that. The promoter section is just a particular sequence of DNA. Part of that promoter is called the Tatum box. It's called that because it's TATA. And on that Tatum box and on the promoter is where these transcription factors are going to connect. Transcription factor is just a protein that is involved in the process of transcription. They all have names. Those names are not important for us in this class. But as each one of them is connecting, you can see here all these different blobs that are proteins. Each one of them is part of what we call the transcription initiation complex. It tells you what it is. Transcription is what it does. Initiation, it starts. Complex, it's several things. The final piece of that transcription initiation complex is this giant protein. It looks like a teardrop here, called RNA polymerase. That is one that you will need to know. RNA polymerase's job is to split open the DNA, as you can see what's going on here. Split open the DNA and create a complementary strand of RNA from one of the strands of DNA. There's another picture of that kind of thing going on here. We'll go back real quick, make sure that we understand. Initiation is this factory being built. Once RNA polymerase comes down, the process starts and it moves to the next stage, which is elongation. Elongation is exactly what it sounds like. Elongation is simply the actual making of the messenger RNA strand, taking those RNA nucleotides, A's, U's, C's, and G's, and adding them to the DNA. And as you can see, this RNA, as it's being made, is just kind of trailing off down here to the side, and this piece of RNA is getting longer and longer as it goes. As it's reading the DNA, it's getting longer. Elongation, very simple. The piece of RNA is getting longer. Termination. Well, termination is exactly what it sounds. This is the stopping of transcription. A stop codon is going to show up the end of the gene, and followed by this long termination signal, AAU, AAA. Not important. You don't need to know that. But as it gets to that stop codon, the RNA polymerase is going to slow down, and it gets that termination signal, and it's just going to stop and break off, and then you have this brand new strip of RNA. Now, in prokaryotes, this is it. In prokaryotes, let me go back to this picture. In prokaryotes, bacteria, that messenger RNA that is made is done. It can go immediately into translation. In eukaryotes, there are 3 modifications that are going to be made to this strip of RNA. Before the RNA is modified, it's known as the primary transcript before it's modified. kind of think of it as like the rough draft. There are 3 modifications that need to be made to the primary transcript in eukaryotes and prokaryotes. That primary transcript is not modified. Here are the 3 modifications. First, a 5' cap is added to the strip of RNA. Remember, we've talked about 5' to 3'. So on the 5' end, a cap is added. This cap serves a couple purposes. It's protective. It protects the messenger RNA in the cytoplasm. The cytoplasm is constantly trying to degrade messenger RNA because otherwise it would build up. And then secondly, there is this 5' cap signals the ribosome. As the messenger RNA leaves the nucleus, it signals the ribosome to connect with it and begin translation. The second modification that's made is called a poly-A tail. It is what it sounds like. It's a run of lots of A's on the 3' end and the adenines, and basically this allows or gives the cytoplasm something to chew on besides the business portion of the gene. The third modification that is going to be made is something that's called alternative RNA splicing. And in this modification, there are these structures called spliceosomes, and the spliceosomes are going to take out sections of the DNA, or this RNA, called introns. On the DNA, there are introns and exons. That DNA is copied. You still have the introns and exons on the RNA. Well, the introns are removed. The introns are non-coding portions of the DNA. And so these spliceosomes come and take them out, and then you have just the exons left. The exons are the coding portion, or the portion of the DNA that is expressed. And so back on this picture, you can see all the introns are removed, and you just have exons. And so the 3 modifications again: 5' cap, poly-A tail, and then the introns are removed by the spliceosomes, and the exons are left. So moving on to part 2, we're basically going to get into translation at this point. A couple of things, just remember, we talked about this just a few seconds ago actually, that peptide bonds are the things that are made between amino acids. Peptide, that's why they're called polypeptides, proteins, because there's more than one peptide bond. And remember, translation occurs on the ribosome. So the messenger RNA at this point is read, and from it a polypeptide is made. Remember, those 3 base units called codons code for that. Well, what brings the amino acid to the ribosome? How does it get there? Well, there's a structure called a tRNA, or a transfer RNA. It does exactly what it sounds like. It transfers the amino acid to the ribosome. How does it know which amino acid to bring? Well, the codon, of course, on the RNA will be matched up with the anticodon from the tRNA. Anti means opposite, and so if you look at this particular transfer RNA, it has an anticodon of AAG, which means that the RNA would have been UUC. So if we go back to the codon chart over here, we see UUC codes for this amino acid. And so this transfer RNA is only ever going to bring that one particular amino acid every time it does anything. It doesn't get anything else. It only gets that one. And so anytime it sees UUC, it goes and gets that amino acid and pops it down. The anticodon is just a place for it to bind with the codon temporarily in order to bring that amino acid. Here's a little bit different look at the tRNA. This bent piece of dynamite is normally how I draw the tRNA, because it makes sense to me. And so here's a picture of that anticodon matching. Again, codon, anticodon. Here's the amino acid. Brings it to the ribosome, and pretty simple. Well, a little bit more about the ribosome. The ribosome is made up of 2 pieces: the small subunit and the large subunit. think too much about that. They are exactly what you think they are. They are, small subunit is kind of the base. This is where the messenger RNA is going to be like it's laying, it's like a table. And the large subunit is like where all the business is going to be happening. It's going to lower down on top of that small subunit to create the whole ribosome. And you can kind of see it on this picture. And so like transcription, when translation begins, it is a three-step process, and it has initiation, elongation, and termination. Again, now one more thing I want to show you about the ribosome. You can kind of see it on this picture actually, so we'll just stay here. The ribosome, the large subunit of the ribosome, also has three sites on it. The E site, the P site, which is hidden by this tRNA, and the A site. The A site is where the tRNAs are going to be added later. The P site is where the actual polypeptide P is being built, and the E site is where the tRNA will exit later. So added polypeptide for P and then exit E. And so those three sites will be important. So for initiation, again, the small subunit is going to bind with the messenger RNA. Remember the 5' cap is that signal molecule. And it's going to align the start codon in the P site right here in the middle, as you can see. Then the large subunit is going to be brought in, lowering, it's going to lower down on top of that, the P, so the P site lines up with that first tRNA, just like you see right here. There's some other stuff going on here, not that important. And so once that everything is ready to go in this picture, then initiation is finished. And so next is elongation. Simple process. Can be silly and long if we made it that way, but we're not. I'm just going to use these pictures here actually to kind of describe this. Just think of the word elongation. What's happening here? Well, it's just getting longer. Nothing crazy. And so here's this first picture. This is kind of where we left off. Notice here comes another tRNA bringing amino acid with it. It's going to come in on the A site. Once that occurs, the amino acid that the newest tRNA brought in is going to attach itself to this longer chain, or the longer chain, excuse me, is going to attach itself to the tRNA and the amino acid that just came in on the A site, as you can see here. And what's going to occur at this point is the ribosome itself is actually going to shift down. That's what you see, this translocation. The ribosome itself is going to shift down so that the structures that are in the P site are now going to be in the E site, and the structures that are in the A site are now going to be in the P site. So that tRNA is going to exit, and now we have this chain being one longer, and the whole thing happens again. tRNA comes in, brings an amino acid. That amino acid is added to the chain. The ribosome scoots over, kicks out that tRNA, and the whole thing is going to keep going. Thousands of amino acids possibly to make this long strain of polypeptide. That is elongation. And then lastly is termination. How does it know where to terminate? Well, we have that handy dandy stop codon. So once it finds the stop codon, rather than bring an amino acid and rather than a tRNA be involved, there is a protein that's going to bind to the stop codon called the release factor. The release factor is going to do exactly what it says it does. It causes the polypeptide to be released, free from its bondage, and headed off into the world of going and doing things. Now, this polypeptide is going to fold up. We've talked about protein folding, primary sequence of a protein, secondary, so forth. None of that's important. It is important, but not for this unit necessarily. That protein is going to fold up, and it could go to the cytoplasm. It could be in the ER and the endoplasmic reticulum. None of that matters, but that protein at this point is done as far as translation is concerned, and it will go fold up somewhere. A couple things to add here. Sometimes more than one ribosome can be translating a single transfer RNA. And so you see that here in this picture. You have multiple ribosomes doing that, just a way to make multiple proteins, saving materials. Part 3, we'll move on. Basically, part 3, we talked about some of the mutations that can occur. Just to quickly back up, transcription, translation. Transcription is making DNA, or making an RNA from the DNA. Translation is making a protein from the RNA. This process doesn't involve any modifications in the prokaryotic cell. In the eukaryotic cell, there are 3 modifications that are made. Now, what happens if there's a problem in the DNA? Any change in the DNA is called a mutation, and a mutation can take several forms. There are point mutations. The point mutation is a change in a single amino acid, or excuse me, a single base. Instead of a C, or up here on the DNA, or here on this RNA, instead of a U, you have a C. That means instead of an A on the DNA, you had a G. There's some sort of problem that can occur. Well, a point mutation can cause one of 3 issues. If the single base change occurs and there's not a change in the amino acid, for instance, looking at this box really helps to show this. Instead of UGU, you had UGC. Well, UGU is the DNA that's, or the, let's go back to the DNA. The DNA here should be ACA, right? Well, let's say instead of ACA, you had ACG. Well, that would still produce a cysteine. It is still a mutation because it's a change in the DNA, but it does not cause a change in the amino acid. This is called a silent mutation, a point mutation where there's no change in the amino acid. If it does cause a change in the amino acid, where that single base causes a change in the amino acid, this is called a missense mutation. You can see here, instead of a glycine, we have a serine. And if that causes an early stop codon, instead of an amino acid, we have a stop codon. That is called a nonsense mutation. All of these are examples of point mutations because they're single bases that have been changed. But they can be 3 different results: silent, no change, missense, 1 amino acid change, nonsense, stop codon. If there's an insertion or deletion of DNA, meaning that something has been added causing everything to shift down, or something has to the left or to the right, and if something has been taken out causing everything to shift to the left, as far as we read here from left to right, that's going to cause all of those codons from that point forward to be different. That is called a frame shift mutation. It doesn't just affect the single part that it was messed up, but it affects all of them after. Just to think of an example that I oftentimes like to use. If you take this sentence, the fat cat sat, well, that makes sense. It's in 3 units, right? What if we added a Q here and still used the same reading frame? Now our sentence makes no sense. Everything has shifted over, still being read in 3s, and now there's nothing. It's nonsense. And so a very similar thing can happen in the DNA because of inserting or deleting a single base or more than 1 base. Nonetheless, it's still causing a shift. Now, mutation can have a wide effect or a really localized effect. If it's a mutation on a somatic cell, that can be a localized effect. Say like a single skin cell becomes mutated. What if a sperm cell is mutated? If a sperm cell has a mutation on it and it fertilizes the egg, and that zygote then has that mutation, then every cell of that new organism is also going to have that mutation. So depending on the type of cell that is mutated, it will have a broader effect.